Today's topic is neurons, type of neuron and biomolecule. So, neuron is of two types on the basis of neuron categorized on the basis of branches and myelin. So, myelin I have discussed previously of myelinated neuron and non-myelinated neuron. So now neuron on the basis of branches. So on the basis of branches, it is of 5 types. So first one is like this, and then it is a polar neuron, a polar neuron. Then like this. So this is bipolar. No, this is unipolar neuron. Then bipolar. So bipolar is like. And then bipolar neuron. Then this is pseudo bipolar. Pseudo unipolar. And then the last one is multinucleated. Like this. It is multipolar. So, a polar neuron in which there is no fixed receiving or giving information pole. Okay, and it is okay, from any direction the information can be in. Okay, from this pole, from this pole, from this pole, from this pole, from any pole the information can be inside.
Can get inside.
And unipolar, in which from this is axon wala part. So from it is cyton, it is the cell body, like there is no dendrite in it, so it is only unipolar neuron from only one end. This is unipolar neuron. There is only presence of axon, not dendrite. And a polar neuron is present in hydra. Hydra is a kind of organism. Unipolar a polar neuron is not present in humans. Okay, it is present in hydra. Now unipolar neuron. So unipolar neuron has no dendrite. It only has axon part, and it is present in embryonic neurons. When our body is on embryonic stage. Okay, when egg and sperm fuse with each other and they form zygote and then embryo. So the embryo which form has a neuron of unipolar neuron. Now bipolar neuron. So it has only one pole of dendrite and one receiving pole and one giving pole. Okay, like one dendrite and one axon. So this is present in eyes and nose. Okay, retina of eyes and nose. Okay. And then pseudo unipolar. So it's look like unipolar. Okay, like he one receiving pole and one. Oh, but it is pseudo unipolar because the body is of upper side. Okay, so it look like a unipolar, but it is a bipolar neuron, and it is found in spinal cord. Spinal cord. Okay. Now multipolar neuron. So this is the true neuron. Okay, and it is present in brain, and most of the neuron is multipolar only. So this has many dendrites. Okay, from many part it can collect the informations, and giving pole is less. Okay, and the axon has also branches. So this is multipolar neuron.
Means multipolar neuron has one dendrite.
No, it has several dendrites. 1, 2, 3, 4, 5.
And this one, what is this called? Axon.
Yeah, this is axon.
So how many axon? One or multiple?
Multiple. 1, 2, 3, 4. Multiple neurons. Now it comes. The neuron topic is completed. Now, neuroglial cells. Okay, so it is divided. Neuroglial cell is non-excitable, and it is also known as packaging cell. Packaging cells. Why packaging cell? Because the neurons, because it packed, because in nervous tissue, there are two types of cell, neuron and neuroglial. So neuron, neuroglial cell packed neurons. Okay, their specific location. So they pack. So they are packaging cell. And now neuroglial cell is of two type, macro and micro. Okay, so macro neuroglial cell and micro neuroglial cell. So micro is the smallest cell, and it is of mesodermal origin. We have I have told that nervous tissue is of ectodermal origin, and but there is acceptance that microglial cells are of mesodermal origin, and it performs phagocytosis. Phagocytosis is cell eating.
What is cell eating?
So the harmful cells, it eats harmful cells. Now macro. So macro is further divided into three types. First one is astrocytes, then oligodendrocytes, and then swan cells. So these are cells only. Astrocytes are the most abundant cell of neuroglial cell. Most abundant. It protects the body from brain and blood blood injury मतलब it create a barrier between blood and brain. Okay, barrier between blood and brain, and it is used for repairing of neuron. मतलब it repairs neuron at the time of injury, brain injury, at the time of brain injury. So three functions of astrocytes. Then oligodendrocytes. So oligodendrocytes are found in PNS, peripheral no sorry central CNS. Found in central nervous system. Oligodendrocytes are found in central nervous system. Its function is to produce myelin sheet. Okay, and Schwann cell is present in PNS, and its function is also to produce myelin sheet. Okay, so this is completed of neuron. Neuron has two parts, neurons and neuroglial cell. Neuron I have discussed, these are types of neuron, and now neuroglial cell. So neuroglial cell are of three types. So this chapter is completed. This chapter was divided into four parts. Animal tissues are divided into four parts. First is epithelial tissue, muscular tissue, and neural tissue, and one is connective tissue. So I have discussed all the four of them.
So now the chapter is completed now.
Okay, so now move to the second chapter, biomolecules. Biomolecules. Okay, so biomolecules are compounds which are made up of carbon. Uh, in living organism only. Okay, मतलब CO2 is not a biomolecule. CO2 is a carbon containing compound, but it is not biomolecule because it is not not.
What is a biomolecule?
बता तो रहे हैं यार 24 मिनट. सो बायोमोलिक्यूल बताया तो कि carbon containing compounds are biomolecules which are present in living organism only. So CO2 is present outside the environment also in the nature. It is a main component of nature, not human body. So the thing like the proteins, the water, which are present in human body. No, water is not biomolecule because water has no carbon in it. Proteins has carbon, so protein is a biomolecule, a carbon containing compound that are present in living organism. The study of biomolecule is known as biochemistry. Okay, and biomolecule now analysis of biomolecule. So it is of organic and inorganic. So firstly I will discuss inorganic. Inorganic in okay. In
What is the
inorganic,
basic difference between organic and inorganic? Basic difference.
जो ऑर्गेनिक कंपाउंड है उनको मतलब वो कर रहा है ना,
English.
ऑर्गेनिक कंपाउंड का एनालिसिस करेगा और फिर इधर इनऑर्गेनिक कंपाउंड का एनालिसिस करेगा, तो उसका डिफरेंस तो
एनालिसिस तो तुम करोगी ना, ऑर्गेनिक कंपाउंड का एनालिसिस और इनऑर्गेनिक कंपाउंड का एनालिसिस. बेसिक डिफरेंस क्या है? व्हाट इज द बेसिक डिफरेंस बिटवीन ऑर्गेनिक कंपाउंड्स, ऑर्गेनिक बायोमोलिक्यूल्स एंड इनऑर्गेनिक बायोमोलिक्यूल्स?
ये तो मुझे कुछ नहीं बताया.
व्हाई दिस क्लासिफिकेशन हैज बीन डन?
मुझे पता नहीं,
ओके,
तो नहीं पता.
Go ahead.
पूछेंगे. ठीक है, फिर इनऑर्गेनिक कंपाउंड. सो इन विच फर्स्ट स्टेप इज टेक अ टिश्यू. Tissue is like we can take a liver, tissue. Then
लीव.
liver, Liver is
So liver.
liver is a
Okay.
tissue. So take a tissue. First is take a tissue. So liver has wet weight. Wet weight.
What is the meaning of wet weight?
The weight of liver with water. Okay, in it.
Okay.
Now dry it. Okay. So when you will dry, the H2O will be separated. Hmm? Now third process is burn. Burn the
Tissue.
tissue. So if we will burn, CO2 and H2O will be removed. The things which we get is minerals. मतलब ashes will be. This process is known as ash or elemental. Elemental analysis. This process is known as ash or elemental analysis. So in which the ash will be left for and in ash it is minerals, metals, non-metal, and metals and non-metal are element. So this will be left out. So these will be the biomolecule. Okay, this is the analysis of compound only, not biomolecule. Compound. So compound. So okay, analysis of compound. Organic compound and inorganic compound. So in inorganic compound, we will found that metals, non-metals, minerals.
As I know, the organic compounds are those compounds where carbon is present, and the inorganic compounds are those compounds where carbon is not present. Am I right?
मुझे नहीं पता ऑर्गेनिक और ऑर्गेनिक का डिफरेंस.
Okay.
तो हो गया मिनरल्स, मेटल्स, नॉन मेटल्स एंड एलिमेंट्स. सो दीज विल बी द रिजल्ट. ओके, नाउ ऑर्गेनिक. सो ऑर्गेनिक में पहला स्टेप भी वही है, टेक अ टिश्यू. सेकेंड है ग्राइंड इट. So if you will grind, it will be like a paste. Now filter it. मतलब छानना है उसको. फिर उसके बाद what will be the result? It will we will left with filter it and retentate. Filtrate is which we get in the vessel, and the retentate is which we get in the cloth from which we are filtering. Okay, so filtrate is acid soluble, cool, and retentate is acid insoluble pool. The particle in which the particles which are present in filtrate are of 18 to 1800 Dalton. Means less than 1000 Dalton. And this is more than the 1000 Dalton. The usual size of the retentate are 10,000 Dalton. This is the size, the usual size, the common size of this. Now, it is clear. These are monomers, and these are polymers. So monomers like polysaccharides, nucleotides, polysaccharides, nucleotides, and amino acids. Okay, and polymers Polysaccharides, the polymer of polysaccharide is monosaccharide. एक second, monosaccharide. Saccharides. The polymer of nucleotides are nucleic acid. Nucleic acid. And the polymer of amino acid is protein. So in retentate polymers are present and in filtrate monomers are present. Now one important point, lipids. Lipids is a monomer. Okay, it is monomer. Its size is um, below 10,000 Dalton. Okay, but it is not the part of filtrate. It's a part of retentate because
कुछ
it
जल रहा
is
है
because it is hydrophobic in nature. Because it is because lipids are hydrophobic in nature. Okay, they uh, they run when water okay, hydrophobic in nature. So when okay, this is of hydrophobic nature. Okay.
जो बोलना चाहती है हिंदी में बोलना
Now micromolecule. So now these are micromolecules and these are macromolecule. In micromolecule, cytoplasm present without cell organelles. And in macromolecule, cytoplasm macromolecule plus cell organelles. Okay, in micromolecule, cytoplasm is present without the cell organelle. Okay, and in macromolecule, macromolecule is present, and with cytoplasm micromolecule, cell organelles are also present. So okay, this is completed.
So today's session is over. Say thanks to everyone.
Thank you.
Say thanks to your viewers.
Hey, thank you.